All right, let's get started. We got a good lecture today. I think lots of really quite applicable things that hopefully you'll be interested in today. So we started chapter 9 last time talking about antimicrobial methods, which normally is kind of, I think of as kind of dry material, but when you're in the middle of a pandemic, you kind of perk up your ears a little bit and maybe become a little bit more interested. So we talked through using heat. We explored lots of different methods to get rid of microbes on surfaces, like drying out the microbes, dehydrating, or adding salt or sugar to pull the water out, or lots of different methods like filtration, as well as the use of radiation, non-ionizing or ionizing radiation. And then we got into some of our chemical disinfectants and antiseptics. So remember, disinfectant is designed to be used on a non-living surface, and an antiseptic is designed to be used on a living surface. Sometimes you can just kind of alter the concentration of a chemical and it can be used for either one. All right, so we started with kind of our low-level antiseptics and disinfectants like soap, meaning it works on microbes that are fairly easy to kill. And then our example of our intermediate chemical, we explored several different halogens like iodine and chlorine. And we have one more intermediate chemical that we're going to talk about, and that's the alcohols. So widespread use right now, the alcohols do. Normally we think about using either ethanol or isopropanol, both as a disinfectant or an antiseptic, usually depending on what the concentration is. One chemical that we skipped over talking about that you'll find in your book are the phenols. We don't talk about phenols that much anymore because they're just a little bit more toxic and irritating to us. Some of the use of the phenols has kind of been replaced with ethanol. So for instance, Pine Sol, that's a cleaner that I commonly use at home because it smells good. Pine Sol used to be a phenolic. That pine oil is a natural phenolic. Well, they actually changed the active ingredient in Pine Sol, and now that's isopropanol. So they didn't change the marketing of it or anything like that, but they did change the active ingredient, and now the active ingredient in Pine Sol is isopropanol. Lysol, that used to be the go-to disinfectant when I was a kid. You just say, oh, Lysol it. That active ingredient used to be a phenolic, a derivative of phenol. And now the active ingredient in Lysol, we don't very often spray Lysol anymore. I usually get those wipes that have Lysol. Okay, that's just ethanol is the active ingredient in there. So widespread use. A lot of you, some of you I saw, use some hand sanitizer when you came in. That's ethanol. The hand sanitizers tend to range from somewhere between 60 and 70%. This particular one that I picked up is 63% ethyl alcohol or ethanol, same thing. This one here that I noticed, it's marketed as a hand sanitizer, 80% ethanol, which is a little bit higher concentration than what you normally see for use on the skin, but somebody must have decided that that was safe at some point. So how do alcohols work? Primarily, they're going to be really good at dissolving membranes. They're also then going to be able to penetrate into those microbes and denature proteins. So kind of work both out on the outside of the cell and inside of the cell. And generally we get between 70 and 95% concentration. So you may think, well, why not 100%? Wouldn't that be the very best? Well, it turns out alcohols actually need a little bit of water to penetrate into those membranes, to work as a surfactant, to kind of pull molecules apart. They need a little bit of that water. So water, in terms of a little bit of dilution, needed for that surfactant action. Actually, kind of like blue. This is blue. Okay, and 100% alcohol actually evaporates really quickly. So it's really typically needs some contact time for all of our disinfectants and antiseptics to work. So you don't want it to actually evaporate really quickly. So again, a little bit of water is good for that as well. It's not irritating to the skin. It's really cheap. So those are other reasons it's so widely used. It is a bit of an inhalation hazard. So that's more so if we use, concentrated alcohol, more of a, if we're trying to decontaminate a room, a whole lot of it, you wouldn't want to breathe that in. What you use in hand sanitizer or spraying it is not enough to be considered hazardous at all. But again, if you do kind of inhale a ton of it, that can affect your nervous system. So again, really high concentrations, it's bad for you. During the pandemic, we switched in lab to using alcohol as a disinfectant on your benches. So when you come in, you spray your bench down. When you leave, you spray your bench down. We used to use bleach, and we switched because then we could kind of spray the microscopes, and basically anything that anybody touches, we could disinfect a little bit better. At some point last year, someone asked the question, well, wait a minute, isn't alcohol flammable? Yeah, it's pretty flammable. So, well, what if you accidentally sprayed some of your alcohol on a Bunsen burner? Seems like that would be bad. So we tried that, and actually the alcohol that we use in lab, I think we use 70% alcohol. It's not, it is flammable, but it's not enough to, like touch somebody on fire. So although I don't recommend you doing that, spraying your lit Bunsen burner with alcohol, again, it shouldn't, cause the whole bench to go up in flames either. When I was a kid, if you really wanted to be naughty, you would take some hairspray and hold it up to a lighter. At that time they had aerosol hairspray, and you could kind of make a flamethrower, but that won't happen again with the alcohol that you use in lab. So, all right, so lots of common uses, both of disinfectant, antiseptic there. Oh, so I have to ask the question, we're not going to talk about the best way to wash hands, but I can't resist bringing up, is it better to use alcohol-based hand sanitizer or to wash your hands? So let's define these. If you're doing both of them correctly, you're going to use enough hand sanitizer to make your hands wet for about 15 seconds. Okay, so again, you need kind of enough of it to cover your hands and enough contact time that it's going to take about 15 seconds to dry out. And if you're going to wash your hands, they say that you should do it at least for 20 seconds. And, when you're scrubbing, you're getting some de-germing action. So, lathering up for about 20 seconds. So if you do those both correctly, is there is it better to wash hands? Is it better to use hand sanitizer? Studies say that it's still best to wash hands. Okay, in terms of getting rid of any pathogens, any disease-causing organisms that would be on your hands. When available, it's best to wash your hands, just because the rinsing, you're rinsing them away. A couple of drawbacks to using hand sanitizer is that it doesn't work as well, or less effective, might be a more eloquent way to say that. Doesn't work as well in organics, presence of organics. So what I mean by that is if your hands are dirty and then you put hand sanitizer on them, that alcohol may not penetrate very well through your dirty hands. Okay, so that's one thing to keep in mind. Also, it doesn't reliably kill non-enveloped viruses. Okay, so we talked about how enveloped viruses out in the environment are really fragile, really easy to kill. Naked or non-enveloped viruses are quite a ways up kind of the hierarchy there in terms of hard to kill. So basically COVID and influenza, enveloped, easy to kill. So yes, hand sanitizer works great against COVID. You get something like norovirus, which is probably the most common non-enveloped virus that we would be infected with. That is not reliably killed by hand sanitizer. So every now and then you get an outbreak somewhere, like on a cruise ship. I think there was an outbreak at Hope College not that long ago of norovirus, which is just really, really super nasty throwing up, really nasty stomach bug. So obviously that's something that you don't want to pass around. May work sometimes, a hand sanitizer, but not reliably. So whenever you have access to it, wash your hands. It doesn't hurt to use hand sanitizer as well. All right, I do have one high-level disinfectant, and this will finish up our discussion of some antimicrobial chemicals that I want to bring up. Again, your book mentions at least one other one, but we'll keep things simple here. Hydrogen peroxide. And we call that a high-level disinfectant because it will reliably kill endospores. So some other disinfectants will kill endospores if you leave on a long enough contact time, but hydrogen peroxide within 10 minutes of exposure will kill endospores. So that's why we consider it a high-level disinfectant. And hydrogen peroxide has been called the closest thing to sort of the perfect disinfectant that we have. Why is it considered really great? Well, it has some advantages, like it doesn't leave any residue, it doesn't damage surfaces, it's really cheap. I think some of those things could be said about the alcohols as well. How does it work? It forms hydroxyl radicals. If you remember, that's a pretty toxic form of oxygen that's going to be grabbing electrons off of other molecules. So it's going to be denaturing proteins, breaking down membranes, causing just a lot of damage in the cell. It can cause damage to the DNA as well. One disadvantage it has is that it's photosensitive. Okay, so like chlorine, remember we talked about you store chlorine in opaque bottles as well. Sorry, bleach. Store bleach, that's also photosensitive. But like chlorine, hydrogen peroxide will start to lose its activity when it's exposed to light pretty quickly. So we always store it in some kind of opaque bottle, or in lab, you're going to be using hydrogen peroxide for a test that we're going to do next week. We store it in these brown glass bottles just to try to keep them fresh for as long as we can. Usually we have to make up that hydrogen peroxide maybe every 6 months or so, okay, or it starts to go bad. So if you remember, I think we talked about this, we produce an enzyme that will detoxify hydrogen peroxide, and lots of bacteria actually can make it as well. It's called Catalase. And that detoxifies hydrogen peroxide just into water and oxygen. Balance my equation there. Okay, so back in the day when I was a kid, we used to very commonly use hydrogen peroxide as an antiseptic. Okay, so when you use it fairly dilute, that's one of the reasons it's so inexpensive. We can dilute it a lot. 3%, we can use it as an antiseptic. So I'd skin my knee or whatever, and mom would spray some hydrogen peroxide on it. That would cause the wound to bubble. Okay, so when our cells and when bacteria cells come in contact with hydrogen peroxide, they make this enzyme catalase to detoxify it, and you end up seeing these oxygen bubbles as a result. That's how you know that the organism's making catalase. So you put hydrogen peroxide on a wound and it would bubble up. You actually have to use enough hydrogen peroxide so it stops bubbling, because then you've overcome the bacteria's capacity to make catalase, and you know that it's going to kill them then. So it's not commonly used anymore because oftentimes maybe people don't use enough. So you have to use enough of it. And you don't want to use hydrogen peroxide to clean out the same wound every day, because it is harmful to our tissues that are trying to regenerate. It will slow down wound healing, and you're more likely to have a scar if you keep putting hydrogen peroxide on it. Okay, so today we more likely are going to use triple antibiotic cream. It's probably just less likely to be misused, but hydrogen peroxide works pretty well as well. If we want to use hydrogen peroxide as a disinfectant, we would use it more concentrated, maybe 30–35%. And again, that's considered sporicidal. We could either use that as a liquid or a vapor if we wanted to maybe disinfect a larger space. We could soak delicate instruments that maybe can't be autoclaved because the blade would be dulled maybe, if we really, again, need that high level of cleaning. So, okay, any questions, anything there from chapter 9 about any chemicals? Again, we didn't touch on all of them that are in your book. I tried to skip over the ones that just really aren't used that much anymore. Okay, so before we jump into chapter 10, our antibiotics, I like to stop and kind of think a little bit about public health, because we're thinking about trying to kill organisms, and now we're switching to thinking about killing them in our bodies. But if we think about kind of big picture infectious disease, how are we doing at treating infectious disease now compared to 1900? And I wish I had, this chart was from the New England Journal of Medicine, and it's a little bit out of date now, right? Best numbers we have here for 2010. So we'll talk a little bit about the past year, what's been going on as well. But if you think about the things that used to kill us most often in 1900 versus in the United States anyway, the things that kill us, more present day, 2010, there's a big difference. So 2010, the most common, and by the way, these are not any figures for you to memorize. This is more because I think this is interesting, and as we're kind of thinking about infectious disease, these are important things. But the things that are going to kill us now, most commonly heart disease and cancer, like again, pre-COVID, before anything else. Some sources I've seen have said, oh, accidents are usually the number 3. They put accidents up a little bit higher here. But other things like non-infectious airway disease. So that's things like COPD, emphysema, cerebrovascular disease. That's, I think, stroke is what that's referring to. We got Alzheimer's on the list, diabetes, nephropathies, so kidney disease. Here's the first time you come into contact with something that's infectious. Pretty high on the list there, pneumonia or influenza. Okay, suicide. Okay, so you compare that with 1900, top causes of death in 1900 were infectious diseases. All right, pneumonia or influenza gets the top billing there. Tuberculosis, gastrointestinal infections. So that would have been things like typhoid or cholera, things that are in water, drinking water, where people used to defecate and then drink in the same water that, without treating it. So this was before we had widespread treated drinking water. Then we see heart disease, strokes making the list. Cancer is a little higher. Another infectious disease here, diphtheria. So, put all those together. What was the life expectancy in 1900 for white men, which I guess that's what we have the best numbers for? Life expectancy was 47 years in 1900. In 2010, that figure was up to 74 years. Okay, so think about what that means. That doesn't mean that you could expect to live 47 years and then die if you lived in 1900. That meant there was still sort of the normal span of life. You had a lot of old people around, but because of all those infectious diseases, the chances of a child dying before their 5th birthday was about 33%. Okay, about a third of children died before their 5th birthday in 1900. All right, so you had old people, right, or elderly, you had a lot of people, kids dying, so that kind of averaged out to life expectancy of about 47. All right, so we've made a lot of progress against infectious disease. Right, until this year. So I was looking for some statistics about causes of death. At least this source, which they get their numbers here from the CDC, so it's probably pretty reliable. They are calling COVID the most common cause of death, at least so far in 2021. So again, infectious disease made a comeback here a little bit. It says that it's not counting accidents not included. That would probably still be in here somewhere. But certainly that's pretty sobering. And a few more statistics, again, thinking about public health and what has made us successful, again, until the last year or two, at cutting down infectious disease. Again, this is looking at the crude death rate for infectious disease. Again, kind of old numbers here. But what things have contributed to us making headway here? Well, we started to understand the sorts of things that cause infectious disease. So this was more like 1860s, 1870s with Louis Pasteur and advances in understanding and Robert Koch understanding what caused disease, advances in hygiene. We started to understand the importance of this, and we've got the government structures in place like health departments. We started treating our drinking water. Again, advances in hygiene alone have really improved our infectious disease rate. We had this outlier, influenza pandemic, 1918. I was trying to get numbers on estimations. In the US, they estimate that about 600,000 people died from influenza, which is, we've just bypassed that number for COVID. Of course, there was a lot less people in the US then, so it's estimated that about 0.5% of the whole US population died from influenza. So again, died down after about 1.5 years there, but there's a huge spike there. But otherwise, because of this, much better hygiene, we were pasteurizing our milk by, the 30s, mostly pasteurizing our milk. So last transmission of plague in the US, penicillin started to be used in the early 40s. That's further helped us win the war against infectious disease. The first polio vaccine, 50s, 60s, started to have a lot more vaccinations in our arsenal, and insist that school children are vaccinated. So the rate of infectious disease is pretty low. There was a little uptick here, probably a resurgence in microbial bacteria infections that were resistant to antibiotics. Some lax, not as many people vaccinating for various reasons. So we obviously haven't won the war, but hygiene, vaccinations, antibiotics, those have been kind of the big three things that are helping us. All right, so as we move forward here, so I like to start with a little bit of history on how the people that kind of were important in discovering antibiotics, as well as what kind of organisms produce antibiotics. So antibiotics are typically biosynthesized. So we'll talk about some definitions as well. Only certain kind of organisms make antibiotics. And we're going to spend a little bit of time thinking about, well, how do doctors decide what antibiotic to prescribe? So clinical considerations there. And probably the most important, we are going to go through about 17, I think, specific antibiotics. So the most important thing that you want to think about is how those antibiotics work. So we're going to call that their mechanism of action. And then we're going to spend a little time thinking about resistance. So before COVID, that was, one of the major things that infectious disease experts were worried about, and they still are, antibiotic resistant bacteria. So how do they acquire the ability to become resistant, and what does that actually mean? How are they evading the antibiotic? All right, so that's where we're going with that. I'm not following quite as closely as what the book kind of goes through, has kind of pages and pages of tables of different antibiotics and how they work, and talk a little bit about, how their resistance and how they're prescribed and things, but I don't follow quite as closely along there. All right, so we have to get straight with some definitions. So we're going to try to use these in the kind of correct sense. Sometimes, again, the word gets corrupted a little bit, or we use it a little bit loosely, maybe in some cases, but we'll try to use these terms correctly. By definition, anything that's considered a chemotherapeutic drug, that's basically any drug we use in the treatment of disease. So we often think of chemotherapy as, oh, these are drugs used to treat cancer. But really that term just means any drug that you use to treat disease. And then a drug that's an antimicrobial. Okay, see if we get it back. All right. All right, hopefully we have it back, our recording. Okay, and then an antibiotic, I have the suspicion that I didn't really indent correctly here. So basically an antibiotic is an antimicrobial agent that specifically a drug that's been biosynthesized. So again, we'll talk about in a second what kind of organisms are going to secrete antibiotics. And specifically, these are drugs that target bacteria. Okay, so that's pretty hard and fast. Like an antibiotic isn't going to have an effect on a fungus, for example. And then we have drugs that are semi-synthetic. So today, we actually can modify parts of that natural product of the antibiotic that's been secreted by the mold or that bacteria. And we can kind of fiddle with it a little bit to give the antibiotic better properties, give it some kind of advantage, work better. So most of our antibiotics today are either fully biosynthesized or they're semi-synthetics. But there are a few that are synthetic. Sorry, hard to read my writing there. There are a few that are considered synthetic drugs. We still consider them antibiotics, even if they have been fully synthesized in the lab, if they target bacteria. Okay. So again, they still target bacteria only. Okay, so for example, we're going to talk about these later, but sulfa drugs are fully synthesized in the lab. We still call them antibiotics because they work only on bacteria. Okay. All right, so discovery of antibiotics. What led up to the discovery? We talked about Paul Ehrlich, I think, on the first day of class. He was a German scientist back around, did his work kind of early 1900s. And he kind of was the first one to really think about treating disease. So he was trying to find a drug that was effective against the bacterium that caused syphilis. And so he very systematically was trying chemical after chemical. And he finally found one that had some spectrum against the syphilis bacterium. Unfortunately, it had arsenic in it. So it did have some use, but arsenic is so toxic to our bodies that as soon as we found something better, then we didn't use that drug. He called it Salvarsan. Again, it was an arsenic containing drug against syphilis. And then there really wasn't any other compounds found for a number of years there until Gerard Domach. Let me make sure I spelled that right. Yep. He was another German scientist, again, in the 1930s. So Nazi Germany, he was working at the pharmaceutical company Bayer. And again, systematically testing a large number of chemicals. And he discovered that sulfa drugs were very effective against a very small number of gram-positive bacteria, but he was trying to find a compound that was effective against streptococcus. And he was successful enough that he got the Nobel Prize for that, but because he was a pacifist, Hitler wouldn't let him go and accept his Nobel Prize, so he got kind of cheated out of that. But these were miracle drugs in the 30s. Any severe streptococcal infection, they were very effective against, credited with saving the life of Winston Churchill, in fact. So miracle drugs, but again, they weren't effective against everything, only a small number of infections. Alexander Fleming, I always want to say Ian Fleming. He made the James Bond films, so different guy. Alexander Fleming, this is him over here posing in his lab in Oxford. He apparently went away on vacation without doing his dishes and came back to his lab and found his petri dishes where he had staphylococcus growing. There was some molds growing in those petri dishes. And so just like this, here's a mold growing, and you can see how it's inhibiting the growth of the bacteria there. Chance favors the prepared mind. I think I said that in lab this week too. But he realized that fungus was secreting something that was effective against killing the bacteria. So he wrote this up in a paper, but I think he isolated the substance and called it penicillin, but he couldn't figure out how to make enough of it where it would be effective at really killing bacteria, even in a test tube. So he wrote it up and then kind of went on to working on something else. That was in 1929. Until right before the outbreak of World War II, Howard Florey and Ernst Chain, and their pictures are right here. Here's Florey and Chain. Howard Florey, he was head of his lab there in Oxford, Actually, Alexander Fleming wasn't working at Oxford. He was working at a hospital in London nearby. But Florey read Alexander Fleming's paper, and he was really interested in trying to figure out, a way to treat bacterial infections. And so he really assembled a huge team to try to figure out essentially an organic synthesis to get to grow up enough of that mold, that penicillin mold, and then to like extract it from the mold. So he had an organic chemist, Ernst Chain, helping him out, and a ton of other people as well. But they were finally successful in mass producing penicillin. And then, of course, that saved lots and lots of lives during World War II, both civilian and armed forces. Okay, so they apparently only 3 people can share the Nobel Prize. So it was Fleming, Florey, and Chain, but again, lots of other people contributed to that. Oops. All right, so where do we get antibiotics from? They are naturally secreted from soil organisms, either fungi or bacteria. Okay, we don't get antibiotics from platypuses or anything like that. Mostly soil organisms, bacteria, and fungi. So a couple of bacteria that have given us lots of medically useful antibiotics, the genus Bacillus. So I've got that pictured up there. If you're in lab, you've used bacilli quite a few times now. And this genus Streptomyces, this is a very interesting soil bacterium, or a whole genera of soil bacterium. They actually grow in filaments, like in the picture here, and they make spores that are reproductive. So they kind of act like fungi, but they're actually prokaryotic cells. Okay, and we get, again, a huge number of antibiotics from various members of that genus Streptomyces. Fungi, some of the famous ones, obviously Penicillium. Penicillium fungi often grow with kind of a greenish tinge to them, but when you grow them up on medium, they're really nice and fuzzy. And then here's another famous genera. We used to call them Cephalosporium. They changed the name to Acremonium for some reason a few years ago. But we get our whole cephalosporin class of antibiotics, cephalosporins, from that genus Acremonium. So just some examples of the kinds of organisms that, again, they naturally secrete antibiotics. We don't really know why. We think that in nature, they don't secrete high enough concentrations to actually outcompete other organisms. So we think it might have some kind of a signaling function, perhaps. As far as I know, that's still kind of a mystery. All right, so what are some of the things that we have to think about, both when we're consuming antibiotics and when doctors are thinking about prescribing antibiotics? So that's what I mean by clinical consideration. And also to some extent, whether the antibiotic ever actually gets made to begin with, if ever gets approved for common use. The number one consideration is that drug selectively toxic? So that means, does it target a structure of bacteria that we as hosts or that humans don't have? Okay, because if something is not selectively toxic, then it's not going to be safe enough for us to use. All right, so when we think about some of the common targets, I'll come back to this slide in a second. Here's a figure from your book talking about mechanisms of action of antimicrobial drugs. Okay, so we're going to talk about some examples of all of these mechanisms of action, except this one we're not going to worry about. That's more for viruses, and we're not going to worry about that right now. A huge class of drugs targets bacterial cell wall synthesis, because of course we don't have cell walls in our bodies. So it's a structure that, again, bacteria have that we don't. So probably half of the drugs that are used now target bacterial cell walls. Another huge class of drugs inhibit protein synthesis. because remember, bacterial ribosomes are slightly different than our mammalian ribosomes. So different enough that, again, we can target that bacterial protein synthesis without harming ours. This disruption of cytoplasmic membrane, you can see there's not a long list here. Bacterial membranes are very similar to ours. So there's huge toxicity issues there. Usually, if we're trying to target bacterial membranes, that's a drug that's only going to be used on our skin. And it's not going to be safe enough for us to ingest. We might try to inhibit some pathway that bacteria have that we don't. All right, some metabolic pathway. So we've got a few drugs that can do that. We have drugs that can inhibit DNA replication. Again, maybe by some enzyme that's just different enough from our DNA replication enzymes, or inhibiting RNA synthesis. So we've got an example of one of each of those. All right. So when I say selective toxicity, again, that's really important. There's always going to be side effects or allergies, but that's a different issue than actually the mechanism of action. Okay, so I mentioned side effects and allergies. That's a big one to consider. How safe is the drug? Allergies, that's an inappropriate immune reaction. Okay, so only some people are going to have that kind of immune reaction. But again, that plays into it. If there's a lot of people with allergy to one thing, then it's just maybe not going to be as commonly used. Toxicity. So there's always going to most drugs have some potential side effects, at least. A really common one is, for example, kidney toxicity. You may have heard that, oh, we're running out of antibiotics, right? We're not making any new antibiotics. We've actually made a lot of new antibiotics in recent years, but most of them are just too toxic. They have too many side effects, so that they're not safe for us to use. So we need antibiotics that are safe for us. Another big issue is disruption of normal microbiota. So, and this plays into our next topic there, spectrum of activity. I didn't give myself a lot of room to write here, but if you take out all your good bacteria, so maybe you're on an antibiotic that's pretty powerful and killing a lot of different organisms, it's going to kill your normal microbiota, what's normally there. And so when you upset that balance of bacteria that's usually there, it can maybe cause certain organisms to overgrow. So for example, you can get thrush. That's what's pictured here. That's an overgrowth of Candida fungus in your mouth, something that a lot of people have normally there, but maybe when you go on antibiotics, again, it upsets the balance in your mouth and that fungus will overgrow. Same issue with vaginal yeast infections. Okay, that can be can be a side effect when somebody goes on antibiotics. Or C. diff, Clostridium difficile. Okay, that's an organism that's in many of us have just normally in our large intestine in small numbers. We go on a strong antibiotic, and again, it cuts down on the variability of organisms in our large intestine. The C. diff can overgrow, cause a really nasty diarrhea. It can be difficult to get rid of. So typically somebody only gets C. diff if they've been on antibiotics for some other reason. Okay, so what kind of spectrum of activity does the antibiotic have? It can be considered narrow spectrum or broad spectrum. So a narrow spectrum drug would be one that would only target a certain kind of bacteria. Maybe say gram positives, or maybe it only will target a very few gram positives. really, that's the ideal thing. If we know what's making the patient sick, we want to give them an antibiotic that will only target, kind of that kind of bacteria. It may seem like, in some cases, a broad spectrum antibiotic is better, one that just kills everything. But then you may have issues with it disrupting the normal microbiota. So if we don't know what's causing the infection, then maybe broad spectrum is best. But again, if we know, we always want to, zero in on it as much as possible so we don't have maybe other secondary infections. Okay. Does the antibiotic work against that organism? We would say, are organisms susceptible is what I mean by efficacy there. And we can test that by doing a Kirby-Bauer test. Okay, so if you're in lab, hopefully that looks pretty familiar because you just did it yesterday or today or this week anyway. What a Kirby-Bauer test is, it's basically a kind of disc diffusion test. So again, if you're not in lab, basically what you do, you take some media and you streak your organism on it that you're trying to see what antibiotics will work against it. So maybe you've got a patient in the hospital who's ill and the normal antibiotics aren't doing any good. So if you can isolate that organism, which may not be easy, you streak it out on a particular kind of media, because this is a very standardized test. We like to use Mueller-Hinton agar. You streak it out, and then you put the antibiotic discs on the bacterium, and then you incubate it and see how well the bacteria is inhibited by that disc. And we usually measure from one side across to the other, and then we kind of see if that measurement, how meaningful that is. Basically, is that considered big enough to be considered susceptible to the antibiotic, or is it resistant? So if you've got growth, say like this one right up to the disc, then that disc isn't having any effect on that organism, and so we would probably call it resistant. Okay. Typically, the larger the zone, the more susceptible the organism is to the drug, but there's some variability there, kind of based on diffusion, how well the antibiotic diffuses through the medium. Okay. Oh, we got to go. Oh no. Okay. So other things to consider. What concentration should we use to treat the patient? Okay. So in order to determine that, we have to know the therapeutic index of the drug. So that basically is the maximum dose of drug a patient can tolerate. So meaning the maximum dose where the side effects don't become too much. Not necessarily that it kills the patient, but when the side effects become unacceptable. So for example, if you are treating strep throat and you give so much of a drug that it makes the patient's hair fall out, okay, that is not an acceptable side effect, right? But if you're giving a cancer drug and it causes that, then that may be considered an acceptable side effect. So, whatever the maximum dose without unacceptable side effects, we call that the toxic dose, and you compare that to the minimum dose that's going to stop the pathogen from growing. Okay, we call that the therapeutic dose. Okay, so let's throw some numbers in here. Say the maximum dose a patient can tolerate would be, say, 100 micrograms, and maybe the pathogen will stop growing at 1 microgram. So then the therapeutic index would equal 100, right? That's a fairly safe therapeutic index, because you have a wide window between where the side effects are going to be too much and what actually works to kill the pathogen. Okay. If you've got, so like, it's hard to look up the therapeutic indexes for drugs. Morphine, for example, has a therapeutic index of 70. So that's considered very safe as well. But as that therapeutic index gets lower and lower, what will work, what's acceptable and what works, if that's getting to, those two numbers are really close to each other, then you have to dose out the drug really, really carefully to make sure you don't get into those toxic doses. So that therapeutic index is going to impact how you administer a drug. All right, so there's different ways to administer antibiotics. Of course, we could, we'll add on to the list here, topical, meaning just on our skin. We do administer some antibiotics that way. You can take them orally, meaning you just swallow them. When you take them orally, in terms of the concentration of the drug in the bloodstream, it never really gets very high. So it's kind of hard to get the concentration up really good if that's what's necessary to give it orally. If you need to get the concentration up a little bit higher, you may need to administer it by shot. Again, that concentration gets up pretty high, but then it dissipates pretty quickly after that. If you need a high concentration for a long period of time, then you're going to have to administer it by IV. Okay, and you can dial in whatever you want. So the administration, that's going to depend on three things here. It's going to depend on the therapeutic index. Okay, so if you have a drug that's very safe, you can go ahead and give that orally. Because you can't always count on people to take their doses at the right time or to take the right amount. You know, I'm guilty of forgetting it now and then, as probably a lot of people are. So if you have a drug that, again, the therapeutic index is low, what if somebody takes 2 pills instead of 1? Okay, then is going to, you know, cause some really serious side effects. So if the therapeutic index is low, you're going to most often be giving it by IV in a hospital setting. You also have to take into account whether it's acid labile. And labile means that it's easily altered. So is the drug easily altered if you swallow it and it passes through the stomach where the acidity is really low? That's a huge consideration. Obviously, if the drug breaks down in the stomach, then you're not going to be able to administer it orally. It has to be given by injection or IV. Also consider how well it's absorbed. So some drugs like penicillin, when we first came out with penicillin, that is actually absorbed really poorly from our small intestine. Only like 25% of the actual drug gets absorbed into the tissues where it needs to go. So, you know, maybe oral is not going to work out very well because you would have to give such massive amounts of the drug. Okay. So those three things all play into how the drug gets administered. You have to consider whether the organism you're trying to kill may be resistant to that drug, and resistance can either be intrinsic, and that means that it just doesn't have the structure that you're trying to target. So, for example, there's a genera of bacteria called the Mycoplasma that don't have cell walls. Right, so if you're giving a drug that inhibits cell wall formation, that's not going to have any effect on the mycoplasmas. Okay, so they have intrinsic resistance, or resistance can be acquired. All right, so how does the organism acquire the resistance to live, say, in penicillin, presence of penicillin? Most commonly, we think that's through horizontal gene transfer. We'll talk more about that. Okay, so it gets a plasmid from another cell that encodes the instructions, maybe to say make an enzyme to to make that drug not effective. Okay, so conjugation probably is the most common. So we'll talk more about that later. And lastly, production. Ideally, it's easily produced and inexpensive. Okay, and then it's going to be in more widespread use. Shane and Flory argued about whether to put a patent on penicillin, and they ultimately decided not to. So that made the drug very inexpensive and could be used widespread. Okay, so our first big group of drugs that I want to talk about are ones that inhibit cell wall synthesis. And they're a huge group of drugs called the Beta Lactams. Okay, so this includes penicillins, a group called the carbapenems, which we're not going to really talk about anymore, but you may have heard of those. The cephalosporins, those are drugs that are in wide use. Okay, these are all called beta lactams because when you look at their structure, they all have this beta lactam ring. And that's if something happens to the drug to break that beta lactam ring or alter it, then they're not going to be effective anymore. And I'll show you why in a minute. So that's kind of a famous structure, a beta lactam ring. Okay. And that might be something, I'm going to show a lot of structures and just kind of point out a few things about structures, but that's going to be something that you would want to be able to recognize, that beta lactam ring. Okay, so how do beta lactams work? We talked about this a little bit in the past when we talked about the growth of cell wall, bacterial cell walls. Okay, so beta lactams actually inhibit peptidoglycan formation by binding to particular enzymes that are necessary for the growth of that peptidoglycan structure. They're called transpeptidases. Okay, we talked about autolysins that will cut the sugar subunits apart, other enzymes that bring in the new sugar subunits, and then transpeptidases actually reform those amino acid bridges. Okay, so this is your book's figure here that explains this. So normally, so here's the normal structure of peptidoglycan. We've got those repeating sugar subunits, NAM and NAG, and this cross-linking of these amino acid bridges. Okay, so normally we've got enzymes that are going to maybe cut between these sugar subunits. New ones get inserted in there, and then the transpeptidases seal up these bridges again when the new subunits get put in, and that's how the cell wall grows. Okay, very carefully orchestrated. But what if the cell, all these green dots here means it's in the presence of some beta lactam antibiotic? Those autolysins are still going to be kind of chopping here those amino acids, those amino acids, those sugars apart. But now the antibiotic, the beta lactams are going to be binding to these enzymes, the transpeptidases. So these cross bridges that should be reformed after the new subunits are put in, you can see over here, are not reformed. Okay, so that basically harms the structure or makes it less rigid, less protective, and then the cell is going to be susceptible to osmotic pressure. So the cell is most often going to be in a hypotonic medium. So cell water is kind of constantly be trying to rush into the bacterial cell. And it's going to take on water, and because the cell wall is not intact, that's going to cause the lysis of the cell. Okay. On a more molecular level, it may help. Again, this is not in the textbook, but if you look at the structure of penicillin, Okay, here's the beta-lactam ring. And you look at the structure of two of the amino acids that are in those cross bridges. And again, and this is in that peptidoglycan structure. And then you superimpose those two structures. You can see they're exactly the same shape. All right, that's important because this D-ala, that's what fits into the active site of the transpeptidase enzyme. Okay, so instead of those amino acids fitting into the active site, penicillin fits in there and what as well instead. Okay, so that's basically a competitive inhibitor. The thing is, though, we don't call it competitive inhibition because it's permanent. Okay, so it's like competitive inhibition because the penicillin fits into the active site of the transpeptidase and blocks the substrate, D-ala, from coming in there. Okay, so it's competitively inhibiting it, but again, because it's permanent, we don't really call it that. All right, so again, that's keeping the transpeptidase from doing its job, from reforming those amino acid cross bridges. Okay, important to remember, this is only going to work on growing cells. If that cell wall is not actively growing, then it's not really going to do any damage. Transpeptidase doesn't have any jobs to do if it's not actually growing. Okay, and again, bacterial lyse because of osmotic pressure, not necessarily because any particular damage is done to the DNA or proteins or anything like that. All right, so as we move forward and I talk about some specific drugs, I'm going to kind of give you a lot of information. It might seem like about some of the drugs. I've tried to kind of pare down things that maybe weren't as important. If you know one thing about the drug, know its mechanism of action. Okay, so for all the beta lactams, it's going to be the same, inhibits new cell wall synthesis. I'm going to talk about things like the how it's administered or whether it's acid labile or not. You don't have to memorize those things. I'm just trying to get you used to understanding the drug in those terms. So I want to get you familiar with understanding, you know, why we would administer by an IV or things like that. So some of those things, again, I just want you to be familiar with the terminology. You should know if there's any particular advantage to using the drug, like, oh, we use this for tuberculosis. This is the only thing that works against tuberculosis, or, you know, these are for common childhood ailments, common uses. That's something to know. And for some drug, there's some big drawbacks to using them. So that, you know, any side effects, that's something that anything that's really notable there. It's something to remember as well. All right. Okay, so we'll start out with the narrow spectrum penicillins. Penicillin G, that's the very first penicillin that's Florian chain, again, mass produced. Why do we call it penicillin G? I think there was several different varieties of it, you know, A, B, C, D, whatever, and penicillin G was the one that showed the best activity against bacteria. So this, again, this was the first antibiotic. Okay, so again, miracle drug, but this was only effective against some infections, against gram-positive infections. But Staphylococcus and Streptococcus, those were a couple kind of notable gram-positive infections. So again, this was a huge help in fighting infectious disease. And it was cheap, especially because, you know, it wasn't patented. So, you know, anybody could produce it. It did reach effective concentration quickly, but keep in mind down here, that first penicillin had to be injected because it was acid labile. So that means that if you swallowed it, it basically was destroyed in the stomach acid. So It had to be injected. And another reason that it had to be injected is because it really, if we swallowed it, it wasn't absorbed very well through the small intestine. Okay, so when it's given by injection, then it did reach concentration very quickly and penetrated pretty well. No big side effects. Ask your grandparents the next time you see them if they remember getting penicillin injections. I know my dad, who was born in 43, remembers very clearly the doctor coming to his house when he was sick and getting an injection in the rear end of penicillin. So, you know, glad that that's not a common thing for us today. Big disadvantage is, you know, we don't use penicillin G hardly ever anymore because the resistance is so widespread. And even Alexander Fleming predicted that would happen, like soon after we started marketing it. And the reason that resistance is widespread is because bacteria often acquire the ability to make an enzyme called Beta-Lactamase. Okay, so let me come back to this slide in a minute. Let me explain a little bit about what Beta-Lactamase is. And this is a figure from your book. Another name for this enzyme is penicillinase. To me, because it acts on penicillin, but I always refer to it as Beta-Lactamase. So again, this enzyme, Beta-Lactamase, is one that sometimes it's hard to remember, like who's doing what. Bacteria produce the Beta-Lactamase. Beta-Lactamase acts on penicillin, already beta-lactam drug. It actually cleaves the beta-lactam ring right here and opens that up a little bit. So now it's not fitting into that active site of the transpeptidases anymore. So that basically just nullifies the penicillin. Okay, so if bacteria can make this enzyme, then they can survive in the presence of penicillin. All right, and again, how do they acquire the ability to do that? Usually they get that by conjugating with another bacterial cell. All right, so the ability to produce that enzyme is become widespread, and that's why, again, we don't really use penicillin G very much anymore. We soon after that actually started growing that penicillium mold on a slightly different medium. And when they gave it a little bit different nutrients, it actually produced a slightly different product, which we called penicillin V. And the only difference in penicillin V is it has an oxygen right here, but just right here. Again, don't memorize a structure there, but that enabled that drug to be able to be taken orally. Okay, so the penicillin V is in some use. I was kind of surprised a couple of years ago when my son had his wisdom teeth out. They gave him penicillin V. They didn't call it penicillin V, but they gave him penicillin just to take prophylactically to keep him from getting an infection. And I was kind of surprised that we use that at all anymore. But again, just that slight change because the penicillium mold was grown on a different substrate. It gave a little different product, one that is able to be still active when it passes through the stomach. Still have to take a whole bunch of it to kind of reach up to the right concentration in our tissues, but okay. All right, and then what typically is referred to as kind of the second generation penicillin. We came out with methicillin in the 1950s. And this is something that's not used at all anymore. So I do have a reason that I wanted to bring it up though. It's just, originally, we monkeyed with a side chain over here a little bit. And that initially was thought that it was going to provide some protection to this beta-lactam ring and make bacteria, slower to develop resistance to it. and it did for a while, but now again, we don't even make it anymore because most organisms are resistant to it. There was a little bit higher side effects. About a third of patients had some kidney toxicity. So that's a pretty kind of high number of side effects to that. So that's another reason that we don't use it anymore. It's acid labile, meaning it had to be injected. So there were some drawbacks. A big drawback, another one, is that this strain of Staph aureus again became resistant to it, and that strain is still around today. MRSA, that stands for methicillin resistant Staphylococcus aureus. So you probably heard of MRSA. It's not as big an issue today as it was maybe 10, 15 years ago. We don't really understand why. But this is a whole, a whole strain of Staph aureus that's not only resistant to methicillin. Okay, that's right. So what? Because we don't use methicillin anymore. But it's not only resistant to methicillin, it's going to be resistant to all the beta-lactam antibiotics. Okay, and there are still a lot of beta-lactams in common use. So, and I tried to kind of figure out exactly, is it resistant to more than just the beta-lactams? I think that it is, but it's kind of information that's always kind of changing a little bit. So I'll have to try to look into that a little bit better. But the point is, a lot of antibiotics that are commonly used, this strain of Staph aureus is resistant to, so it's just difficult to treat to get better. Often manifests as a skin infection, but it can be, internal after a surgery, and then that's a huge problem. Okay, so while we're talking about the beta-lactams, and I have a few more that I want to talk about, I did want to mention that allergies to penicillin is another problem. It's estimated that about 10% of us in America have allergies to penicillin. So again, this is an inappropriate immune response to penicillin that we metabolize. So it's the metabolized product that some people have actually a specific IgE antibody reaction to, that can cause things that are really just kind of uncomfortable, like a rash or hives, and it might not be anything too serious. One of my colleagues upstairs on the third floor of Henry had hives for 9 months after he had penicillin. So if you've ever had hives before, like for a few days, like 9 months of that would absolutely drive me crazy. So it can be severe. Most people probably just had a little rash when they were a kid, and then, okay, now they can't take penicillin anymore. But it can lead to more serious reactions like anaphylactic shock. So that means that your blood vessels get so leaky that you're not getting enough oxygen to your organs. And about 300 Americans die every year of a hypersensitivity reaction to penicillin. So because I had a rash when I was like 4, now I can't ever have penicillin again or any beta-lactam antibiotic. And, I went to an allergist to try to get them to test me to see if I was really still allergic, and they wouldn't do it. He said, well, we would do that if we had to. So meaning, if I was at death's door or something, they might do that. But the so what is, like my daughter over the weekend had an ear infection. So she got some amoxicillin at Meijer. It was free. All right. Like those of us that are allergic to penicillin, when we go get an antibiotic, it's like $100. So it'd be nice to know if I really was still allergic to penicillin or not. Okay. So we will move on with more antibiotics on Thursday. Have a good day.